of antibodies to allergens and the role of TFH cells, which, as you will see, are not quite what we expected. So we're going to talk about this paper that just came out earlier this year. And I'm going to start by giving a little bit of background about the antibodies that we're going to be discussing, because this is the main focus of my lab, is understanding that DC T-cell, B-cell interaction that generates different types of antibodies, good antibodies, bad antibodies, however you want to call them, protective, pathogenic. So in food allergy, one of the antibodies that's thought to be potentially protective is IgA. And so what you see here is a recent perspective that came out in Science talking about different types of antibodies to allergens, in particular to food allergens. And obviously in allergy, we worry a lot about the IgE antibody because it's the antibody that induces mast cell degranulation and a lot of the pathology we think about with anaphylaxis. But there are counter antibodies to the IgE response. In humans, IgG4 is one of them. And in mice and humans, IgA is also thought to potentially block the IgE response. So you can see that's highlighted here, in particular, highlighting its role in the gut. Remember, IgA is a mucosal antibody. So it's in your lungs, it's in your gut. And it's been shown to have a very important role in those mucosal sites to other types of antigens. And so we're going to come to that in a minute. Its role in food allergy, though, is actually a little bit unclear. So let me give you a little bit more information just about IgA. So as I mentioned, it's a mucosal antibody. In the gut, it is the dominant antibody there. And actually, when you find it in the gut in secretions, it actually has a unique form where it's a dimer. And it has a particular component called the secretory component that helps it get out into the lumen of the gut. So this is the inside of your gut. And that's where bacteria are. That's obviously where food antigens would be. And it's actually transported there actively. So it's stabilized by a number of other molecules like the J chain that make it in this dimeric form. And just work from our own data corroborates that this really is the dominant antibody isotype in the gut that we should be thinking about. And it's been studied actually quite extensively in its role in regulating the microbiome in the gut, also in neutralizing toxins. And there it's been shown to have a very important role in dictating what type of bacteria are in your microbiome. So potentially blocking the negative ones, the pathogenic bacteria from getting in, and shaping the commensal bacteria to have a homeostatic or healthy balance. And that's been fairly well established. There's been a number of different cells and molecules that have been shown to be important for inducing that response and making sure that that balance is correct. And also for toxins in terms of blocking their ability to get into the body and cause disease. Yet I put food here with a peanut or the question mark because there's not really a lot of literature on IgA to food antigens. Not how they're induced, not what they do. One of the models that's out there, if you look at a textbook and it mentions food allergy, it would go something like this, where you have antibodies, the brown double antibodies here are your IgA binding peanut. Again, this is in the lumen of the gut. And if you lack this antibody, the idea is that this dangerous allergen, peanut, gets across and induces an IgE response and potentially an anaphylactic mast cell degranulation response. So this is a very attractive model. It very much is consistent with what we know about IgA to toxins and bacteria. Problem is there's almost no data to support this model. It's actually also mechanistically quite difficult to explain and to understand how it's induced. And so we're very interested in this question. And so we set out to first understand, well, how is IgA at all induced to food antigens? And is it the same or different than those that are generated to bacteria? And actually, as I'll show you, the types and nature and duration of antibodies, of IgA antibodies to food seem to be different than bacteria. So there was reason to think maybe this would actually be a different response. So we just started first by characterizing a group of healthy adults. So these are non-allergic people to see, can we even detect IgA to peanut? And this is looking in stool samples. So these are trying to reflect what's happening actually in the gut, not in the blood. In humans, IgA in the blood and the gut can actually be slightly different. We have 2 IgA genes, and their relative abundance in mucosal surfaces versus the blood are actually slightly different. So, but this is just looking at total IgA to peanut, and you can see that most people we sampled actually made an IgA response to peanut. We actually could tell that this IgA was quite specific to peanut because we could block the ELISA results using peanut itself, but not other kinds of food antigens. And so this argues that there's a high degree of specificity, which is actually different than what people have seen for IgA to bacteria, for example. Those IgAs often can be what are called cross-reactive and broadly binding different kinds of molecules. The other thing that was really striking is we sampled 3 people over the course of about a year. And so you can see in these 3 different colored lines, these are each 3 different individuals, that people who make a lot of IgA to peanut kind of stay high, and people who don't make a lot of IgA stay low. And so this argues to a really durable, somewhat maybe high affinity type IgA response, really arguing that the antibody really is targeting peanut. But the question was why? And actually, we also checked a couple of other food antigens. In this case, we're taking egg white. And again, although we had more people who did not respond to egg white, most of the people we sampled had IgA in their guts to egg. So the question that we're really trying to tackle in this paper is what's stimulating this response. So if you look in the literature again, or in reviews really, how this is all induced, there's some really nice theories out there, but actually there's not a lot of scientific experimental data to support it, but these are some of the dominant theories. So in this really beautiful picture from a review a couple of years ago, here's an example of a peanut coming in and potentially inducing what would be called quote unquote a tolerogenic dendritic cell. And this dendritic cell is then going to the mesenteric lymph nodes, so the gut draining lymph nodes, and inducing a tolerogenic response. And that includes both T regulatory cells. So that's a fairly well worked out process. This is what we might call oral tolerance. And Tregs are a very important piece to that, oral tolerance. But then you can also see down here on the left-hand side, these Tregs are inducing a B cell response that induces IgA. So again, there is some experimental data suggesting that Tregs can induce IgA in general, but most of this process that's shown here is hypothetical. We don't actually know. And so one of the questions that's kind of intertwined in this project is really, is IgA part of the oral tolerance mechanism? And if so, does it have a tolerogenic function? And so I think one of the best ways of trying to address that is by understanding exactly how it's made and then perturbing that. You know, so in the world of understanding IgA to bacteria, there's a lot of different pathways that can get you to an IgA to that bacteria. And some of them are T dependent and a lot of them are actually T independent. So a very different way of getting a B cell response. And, you know, there'd been a number of papers out there suggesting that many of the T dependent IgA to bacteria are actually Tfh dependent. But as I'll show you, that actually had not been demonstrated experimentally. So we decided to try to test this out. And then as part of this, kind of the immunologic site of induction has also been postulated and actually mostly disagreed upon, but that's usually been done for work that has looked at IgA to toxins. So again, these are all open questions. And actually based on the work I'm going to show you today, we were only able to answer some of them. We're still working on a lot of these questions. So to start with, we went to a mouse model. So my lab utilizes the back and forth between information from clinical data, like the one I showed you in the first figure, going to mechanistic animal models, and then hopefully going back to validate in human samples. And that's what I'm going to show you today we did. So we started by doing a basic exposure model, very much like many of the oral tolerance models that have been used for mice. So here we're feeding peanut daily to mice and seeing whether or not we can get the same kind of IgA response that we observed in our healthy human cohort. And so after almost 2 months of feeding these mice, we got very little stool IgA to peanut, which is shown here after day 42. And even if we started mice in infancy, so essentially as they were being weaned, we started exposing them to peanut, we still didn't get a lot of IgA. Further, we couldn't really do something to the peanut to try to make it more immunogenic, meaning get it to induce more of an IgA response. One of the things that's postulated to make peanut such a potent allergen is its roasting. So obviously in the United States, a lot of people eat peanuts, they're actually roasted. And it's thought that maybe that modifies it in some way that makes it allergenic. But we tried that and it doesn't seem to change the IgA response at least. But further, when we looked a little bit more at these antibodies that were being generated with this daily kind of chronic exposure, first of all, when we knock out all T cells, so these are using C2TA knockout mice, so they don't have CD4 T cells, they actually didn't really change the IgA response. And then further, when we looked at the mice that actually did make a response, although it was low level, we couldn't really find a pattern like I showed you with the human samples where we could block it with peanut, but not with other allergens or antigens. It kind of looked like it was all over the place. So this looks very much like what has been shown for a lot of the bacterial reactive IgA, where it's binding to a lot of different motifs, not even bacteria, but other widely expressed motifs. So it looks cross-reactive and not very high titer. So again, this doesn't look like kind of what we had seen in our human subjects. So we turned to another system. And so these data really raise the question of either, so mice are just not mimicking what we're seeing in humans because they have different types of gut responses, and that's very feasible. They obviously have a lot of differences in terms of their environment and also in terms of their immune system. Or we are missing something about the mouse system that would help us explain the human data. And so we decided to tackle the second question to see if we could induce an IgA response that more mimicked what we saw in the human samples. And so we turned to what has been classically used as a food allergy model in mice. And this involves giving peanut with what's called a mucosal adjuvant cholera toxin. It actually is the toxin from Vibrio cholerae. So it induces, in humans, massive diarrhea and can be actually life-threatening. But it also, by itself, can act as a potent adjuvant when administered orally. And so we use this in combination with peanut, and that's what this little flame symbol here means, is we're giving an adjuvant now to initiate an innate immune response, typically driven through dendritic cell activation, to initiate an adaptive immune response. And accordingly to what the peanut allergy models would say, we induced a very strong IgE response. However, we look to see, do we induce other types of antibodies as well? And indeed, we induced an IgA response, a very strong one. So the scale here is the same as the previous slide that I showed you. And so we're logs order higher than what we just saw with daily feeding. Further, the nature of the antibody is quite different. So now we very much mirror what we saw at the very beginning in the human samples, where we can block this antibody with peanut, but not with other food antigens. This was true for peanut, this was true for egg. We used ova to do the same thing. These antibodies are not cross-reactive, so you can't induce an ova-specific IgA and have it bind to peanut. They really are separate. And we induced not only a gut IgA response, but we also could see some in the serum as well. It wasn't quite at the same magnitude. In parallel, actually, we also saw IgG come up, and this is an antibody that in the gut is not thought to be a very dominant antibody. In fact, IgM is one of the other major isotypes that you see in the gut because it's also transported actively in the way that I showed you at the beginning for IgA. But we do see a lot of IgG. Now, these scales are using arbitrary units, using standards that we developed in our own lab. They can't be directly cross-compared. So this level of IgG is not the same as this level of IgA. We can tell that indirectly by doing limiting dilution ELISAs to try to check the OD value, but this is only an indirect quantitation. In general, most of the antibody that is made to peanut is IgA, both in our mouse samples and our human samples. But you can see it's a pretty strong response. And really surprisingly, you don't get any IgM. Maybe not surprisingly, you don't get any IgE either. That IgE response really is different. It's really in the serum. We'll come back to that in a little bit. But I think an open question in the field is, what is this IgG doing and how is it getting into the gut lumen? It doesn't get transported through the PIGR receptor the way IgA and IgM do. I'm going to leave that question aside. That is an open question that we are still actively thinking about, but we don't have an answer for. The other thing that was really striking in my mind is that you could get the same kind of IgA response if you gave adjuvant just the first time the mouse was exposed to peanut, but stopped giving it subsequently. So in many of the food allergy models, we need to give repeated antigen in order to get an IgE response. And we saw the exact same thing for IgA. You really needed to have multiple rounds of antigen exposure. And there's some theories as to why that might be, involving how the B cells get activated. I'm not going to go into that right now. But if adjuvants are needed, they are typically needed, as I mentioned, for that innate immune activation to get T cell priming. So like a dendritic cell to get activated in order to induce naive T cell priming. And so we hypothesized, and it turned out to be true, that you'd only need cholera toxin really at that first exposure to induce that T cell response. And then the subsequent responses would just need antigen to boost the B cell response. And that's exactly what we saw. They were essentially overlapping responses. And that was true for E as well, and the IgA response we saw in the serum. So this obviously leads to the question, getting to the question about the T cell response. So the first thing we checked was whether or not you needed CD4 T cells at all. And unlike that response that I showed you at the beginning, where the kind of very low level cross-reactive IgA was not T cell dependent, here this is completely T cell dependent, CD4 T cell dependent. The type of antibody we get is also quite long-lived. So if you stop giving peanut, you can see this antibody out for even months later. So this argues to a real T cell dependent B cell antibody response, and would argue really for a TFH response. So I don't need to go into any more introduction than this, because I think Shane just did the best job that could be done to introduce these cells. But obviously we think a lot about the induction of B cell responses driven by different types of T follicular helper cells. And a lot of the times we think about this in the images Shane showed you in the germinal center. And these are the kinds of interactions that we think about driving these types of antibody responses. And as I mentioned, this kind of interaction had already been postulated to be necessary for IgA responses to things like bacteria and to toxins. And so that's why in many of the reviews and models, TFH cells have been incorporated into this interaction to drive IgA in addition to these other isotypes. I will point out, because this is my personal bias, IgE is missing here. It's also necessary for IgE as well. So we just did the straightforward experiment, which was to delete the TFH cells and see if it altered the IgA response to peanut. This had been, like I said, this has been postulated to be a cell type necessary, but it actually never been directly demonstrated by modulating the TFH cells. And so we did actually see induction of TFH cells, both looking at BCL6, but also looking at PD1 and CXCR5 staining. This is looking in the mesenteric lymph node. The immunization that is associated with IgA induces a TFH response. Interestingly, the potentially counter-regulator of this response, T follicular regulatory cells, so FOXP3 positive TFH cells, were decreased. TFR cells have also been postulated to be one population of T cells, though, that drive IgA, in part because TGF-beta is actually a very potent switch factor for IgA, and these cells in adoptive transfer experiments had been shown to promote IgA. And so we were characterizing both of these and saw this interesting but differential response. But to us, the real test was to knock them out and see what happens. And so we used a CD4 Cree BCL6 floxed mouse, essentially similar to what Shane just showed you. And indeed, you can very effectively delete TFH cells in these mice. They don't have germinal centers. They don't make effective TFH-dependent antibody responses. You can see here that the germinal center B cells by flow, also by IF, which I'm not showing you, are really ablated. And what we saw, though, was completely shocking, which is that, yes, we didn't change total IgA, and we didn't really expect to maybe change total IgA in the gut, but we actually didn't change peanut-specific IgA at all. And that was quite shocking. We looked at this both in terms of using our ELISA or by taking out antibody secreting cells from the gut or the mesenteric lymph nodes and re-stimulating them with peanut to see how many antibody secreting cells we could get. And again, there was really no difference. So this argued for a TFH and potentially germinal center independent mechanism for IgA induction. But importantly, when we looked at other isotypes, which we know are TFH dependent from previous work, like IgG1 and IgE, those responses were dramatically impacted. They were really TFH dependent. And so you could actually visualize this pretty clearly by just looking at the B cells in the mesenteric lymph nodes. And here we're looking at the plasmablasts. And you can see that the IgG1 plasmablasts in that lymph node are really just completely gone. But the IgA response, if anything, was higher, is what was left of the B cell response. So you really weren't hurting that IgA induction. And so this argues for two separate pathways for food-specific antibodies for these different isotypes. And that raises a whole host of questions in terms of differential function, how these B cells are being induced, but that the T cell response is actually distinct. It also led us to think about, well, where might this be happening? So this is some immunofluorescent images from the mesenteric lymph nodes in these mice. And you're seeing here on the bottom in what's labeled D, you're seeing TFH deficient animals, and in the top you're seeing control animals that do have TFH cells. And in both cases, we could find IgA B cells in kind of this TB border area. And we know that IgA staining is fairly specific, because if we use AID knockout mice, which really depletes the ability of the B cell to make IgA, we don't see IgA staining. And if you put this all together, and I know this is a lot of colors on the right-hand side, they're all separated out, you can really, in a wild-type animal, try to visualize where this IgA response is happening. And it looks like it's really at the periphery outside of this germinal center. So we went on to test whether or not these TFR cells were also necessary. And that's an important population to test, as I mentioned, because of the work that had been done showing TFR cells were important for certain types of IgA. And so again, we can do this using a flox mouse for BCL6 and deleting it in FOXP3 positive T cells. And so you really do efficiently ablate the TFR cells, but you don't change the peanut IgA. So again, arguing that these two populations that are related, but really aren't necessary for this induction. However, we went on to look at CD40 ligand dependent mice and humans. So the mice are at the top and the human samples are at the bottom. CD40 ligand, remember, is a very important effector molecule that T cells, Tfh cells in particular, use to license B cells, so through cognate interactions. And when you delete this molecule, you completely lose your peanut-specific IgA. This looks almost like a T cell knockout mouse. So it argues that the same kind of cognate TB interactions are operational here, yet not happening through a traditional, potentially, TFH type response or TFR response. And importantly, in the human samples, we could see the same thing, although CD40 ligand seems to be, and interestingly, much more important for all IgA in the gut, not just peanut-specific IgA, where that does not seem to be true in mice. We don't understand this difference yet. So to summarize this, and I'll show you the model that we are working on, chronic peanut exposure really doesn't lead to much IgA. So if you have food antigens coming in, potentially, for example, like a oral tolerance type model, they don't seem to be activating much of a B cell response. You get a low-level B cell activation that makes a cross-reactive antibody that seems to bind a number of different antigens. This looks a lot more like the IgA that's been described to commensal bacteria. This IgA, however, is T independent, so a very different pathway. And so it kind of questions this idea that IgA is part of the oral tolerance pathway. And it leads to a very difficult question that we are trying to actively address, which is what role, if any, does IgA play in mitigating allergy in terms of the food-specific IgA? Now, in contrast, when you have an inflammatory reaction induced by an adjuvant or some kind of innate immune stimulation, you get then very high levels of peanut-specific IgA. And that's shown here. You have inflammation coming in with your antigens. This induces a T-dependent B cell response that is CD40 ligand dependent. So the same type of rules that we think about between a TFH and a B cell are occurring in the secondary lymphoid organ, which we actually don't know which one that is in the gut, but is inducing a very strong B cell response that's highly specific. It's not binding to a lot of other antigens, and it's really neutralizable by peanut alone. And so an open question that we're still really actively working on is, what's the nature of this T cell? But a second question that gets raised is, well, what is the adjuvant that's potentially inducing the same kind of response in humans, since we saw a very highly specific, high titer IgA response to peanut in our healthy individuals? All right, so with that, I will stop. I really want to thank my entire lab. We all tend to work together on projects, and this really was a group effort. This was a lot of work over many years. But I just really want to highlight Bian Zhang, who's shown here. This was at one of our lab retreats recently, before when we can actually all get together without masks on. She was a graduate student in my lab. She graduated. She's now doing a postdoc, but she really started this project from the ground floor. She got the models working and really stuck through figuring out all the nitty-gritty of these different aspects of this response, despite it kind of flying in the face of what had been out there in a lot of the published literature, which was hard. We took a lot of wrong turns and spent a lot of time making sure what we saw was right, because it was so different than what we expected to see. She was helped by an incredible team. We call them the IgA team. Jake Gertie, Elise Liu, John Anthony. They've all been helping. They've been joined recently by Emily, who's just recently joined the lab, and everyone else in the lab who's really been working together to understand this surprising and interesting question. And I really do want to also point out, I've got a number of collaborators who've helped us understand all of this, and some incredible funding from two groups that took a chance on us working on IgA to food, even though nobody really understands the role of IgA to food. And that's the FAIR Foundation with the Rickless family and the Stanford Sean Parker Center, in particular through Carrie Nadeau there. So with that, I will stop and I'd be happy to take any questions.
Great, thank you for the talk, Stephanie. That was really insightful. If there are any questions, you can direct it to the chat. All right, we have a first question from Rod Rahimi from Massachusetts General Hospital. Thank you for the very nice talk. In your mouse experiments, have you used other adjuvants besides CT? Do you see similar biology with alternative adjuvants?
Great question. It is something we are actively working on. In the paper, we showed that other kinds of allergens or immunostimulants, like Alternaria, which is a fungus extract, can actually also induce the IgA response, but we're not sure if the nature of the IgA is identical. We're still trying to figure that out. I will tell you, we have tried a lot of adjuvants that are either out there and thought to potentially be mucosal adjuvants, or ones we just are trying, and not a lot actually induces IgA. So there's something very unique about this process that's highly regulated, and I think it's a fascinating question. So to be continued. Hopefully you'll see that soon.
Great. We have a second question from Jerry Lin. Would you expect that CT is sufficient to induce peanut-specific IgA in germ-free mice? Any role of endogenous microbiota-derived mediators?
Yeah, nice question. It's a little bit hard to know, but CT has some very potent effects even directly on dendritic cells. This is work from Niles Lickey's group and already. So they've done a lot of this work, and CT is very potent as a molecule itself. So I don't know that you actually need microbiota as well, but it's hard to know. The experiment would need to be done.
Great. And we have a question from Jose Carlos Crispin. Congratulations for your work. IgA deficiency is associated with autoimmune disease. Is the same true for food allergies? TGF-beta is known to be important for IgA switch. Could you speculate on the identity of the IgA helping T cell? Would it be the source of TGF-beta?
Lots of questions in there. They're all really good ones, and they are all ones we are working to understand. So the IgA deficiency, IgA deficiency, just for everyone else, is a relatively common primary immunodeficiency. About 1 in 200 to 1 in 500 people actually lack IgA. And so it early on had been thought that maybe IgA might predispose to food allergy. And so some of the studies have shown that there are some aspects of food allergy that might be enhanced by IgA deficiency. But in general, the answer is no. Those patients are not predisposed to symptomatic food allergy. So that is in part why the question about the role of IgA to food is still open. I'll tell you, when we work on this, we do get a lot of questions about why we're even working on it, because many allergists don't think it has a role. But we make a lot of this antibody, so I think it has something, it has to do something. Now the question is what, and that's a hard question to answer, but we're developing the models to do that. Regarding your other questions, we have speculated a lot about the type of T cell that might be relevant. The other one primary candidate that we thought about was regulatory T cells. I showed you data on T follicular regulatory cells, but just garden variety Tregs have also been shown to be important for IgA to other types of target antigens. And it makes a lot of sense because they make TGF-beta, as I mentioned, and TGF-beta is an important switch factor for IgA. And so we actually induced Treg deletion and tried the same model, and also didn't see a defect. So we don't think it's Tregs. So we're still working our way through it. If you have thoughts, and you want to have a discussion offline, happy to keep postulating, but it's only at that stage right now.
Okay, we have another question from Eugenio. Thank you, Stephanie, for the great talk. Is there any evidence that indicates TLR-dependent mechanisms associated with the IgA response? Some data indicates that TLR signaling could activate B cells in an independent manner of T cells. Could you get IgA production in the absence of CD4 T ligand by giving a strong TLR signal?
Okay, yeah, so nice thought. The thought here is that B cells express many toll-like receptor ligand receptors, so pattern recognition receptors, TLRs, and can get activated in that way. So that can bypass the need for B cell, T cell, CD40 ligand interactions and induce a really strong antibody response. So it tends to be obviously T independent, but also of a different flavor of antibody. And that's why I spent so much time trying to understand specificity and titers and T cell dependence in our work. And so those tend to be more cross-reactive and lower titer than what we're seeing. So I don't think so, but it's possible. We definitely have not ruled that out.
Great. So we have two questions from Marco Aurelio Tapia. One of them is also a question that I had. Have you measured IgA in stool of the BCL6 deficient mice after several weeks of discontinuing peanut orally? I think you had showed a long-term graph of the peanut IgA. So had you done something similar for the TFH mice?
Oh, I should have explained this and I didn't. The TFH mice, we sampled at the peak of when we typically see an IgA, the peak of the IgA response, which is usually about 6 weeks, so 6 to 7 weeks after immunization. So all of that data later on, when you just see one single time point, is at a later time point, like the 6 to 7 week mark. Do you mean later than that to see if it ever diminished? Is that the thought?
Yeah, I guess
Yeah.
like how long it took the IgA was able to persist in those mice.
Yeah, so Bien had the same question. She thought about that and did actually follow them out for some time after discontinuing. And they have a slight reduction, just like the wild type mice have a slight reduction, but it still looked pretty similar to what we saw in the wild type mice. It's a good thought. It's a really good thought.
And the second question from Marco was, have you considered using non-SPF mice to mimic the relatively dirty microbiota that humans have in comparison?
We have, and we really want to do that experiment. I think it's a great idea. We're trying to figure that out right now. Thank you.
Okay. I think we can do one more question, just lastly from Ariel. Thank you. Knowing the difference between the gut environment and the lung environment, have you investigated or could you possibly see this antibody B cell responses in the lung mucosal surfaces to the peanut particles when it is inhaled versus ingested?
Yeah, that's a great question. I wanted to do it. We haven't had the bandwidth to do it. We have a lot of projects going on, but we do want to tackle the lung. I think it's a really important comparison.
Okay, well, great. Thank you for your talk.
Thank you all for the great questions.
Yep, I'll hand over back to Valencia.
Okay, thank you. So I'm honored to introduce our third speaker today, Dr. David Hildeman, who got a bachelor's degree in biology at University of Wisconsin, followed by a very productive PhD in microbiology and immunology, studying T-cell responses to vaccines. He then did a successful postdoc with Philippa Marek and John Kapler at Howard Hughes Medical Institute, where he looked into molecular mechanisms of T-cell death. And since 2002, he got a faculty position at the University of Cincinnati, where he started his own lab that has importantly contributed to the understanding of the mechanisms that mediate T-cell tolerance. And without further ado,
Oh, hello? Can everybody hear me?